All right, so let's talk now about the central dogma of genetics. A dogma is like a core idea. So the central dogma of genetics is, here's the core idea about how genetics work. It describes the flow of genetic information in cells from DNA through transcription to messenger RNA, and ultimately to translation to proteins, where those proteins determine the structure and function of an organism or a cell. So the central dogma also includes the process of replication, which we talked in our previous video, that happens in the S phase of interphase and creates identical copies of the entire genome. All living things create proteins using almost this exact process. This is yet another example of conserved traits found across all life and common ancestry across all life. Here we see the central dogma. So DNA being used as a template for transcription to RNA. RNA uses the directions which flow out to the ribosomes. The ribosomes use those directions to put together amino acids and make proteins, and the proteins ultimately determine traits or the structure and function of living organisms. And again, right here, we see replication included as part of that central dogma. So there are a couple of exceptions to the central dogma. They're non-living things. Retroviruses like HIV, for example, and influenza, and even hepatitis B, they don't have DNA. They're RNA viruses. Their only genetic material is RNA. So these viruses inject their RNA into a host cell along with a special enzyme called reverse transcriptase. This enzyme uses RNA as a template for making DNA. That DNA is then ultimately incorporated into the host cell's DNA. It will later be transcribed back into RNA and used to make proteins. So again, that's an exception. They have to start with RNA. There are also some virus species so primitive that they only use RNA and go straight to proteins. They never developed DNA or the ability to work with it. And then finally, we have these things called prions, which are self-replicating proteins. So a protein can cause the replication of that protein. We're still learning about prions, but we know that they're involved in especially a lot of nervous system diseases like mad cow disease, Kuru, and Creutzfeldt-Jakob, CWD, and also chronic wasting disease in deer. So proteins, their function, or the only function of DNA rather, is to code for proteins. That should tell you how important proteins are. They make up much of the body structure, and they're responsible for most of its functions. Back in 1941, Beadle and Tatum proposed the one gene, one enzyme hypothesis. And this idea was essentially that each gene codes for a single protein or single polypeptide. Now we know today that's not exactly true, but we'll talk about some exceptions of that later. Before proteins are made, we know that the DNA is used as a template for making messenger RNA. We call that transcription. Transcribe means to rewrite or to copy something. Messenger RNA, why it's necessary is it's small. It can leave the nucleus through pores in the nuclear envelope, and it can go out to the ribosomes, attach to them, and essentially it can function as a disposable or portable copy of the protein building directions found in the DNA. We know DNA can't leave the nucleus, and that the ribosomes are found either attached to the rough ER, which is outside the nucleus, or freely floating in the ribosomes. Transcription is the first step in gene expression. And unlike replication, where the entire chromosome is copied at the same time, transcription occurs and is controlled on a single gene by gene basis. The process of transcription occurs in three main steps: initiation, chain elongation, and termination. So let's talk initiation. During initiation, a group of proteins called transcription factors attach to the promoter region found near the gene. So each gene or a group of genes contains this promoter sequence. It's essentially a section of the DNA, oftentimes very heavy in TA nucleotide sequences, sometimes called the Tata box, that essentially signal to enzymes, hey, this is the beginning of the gene. Once those transcription factors bind, next, the main enzyme of transcription, RNA polymerase, attaches to the promoter. It actually assembles the new RNA molecules. Sort of like helicase during replication, it first separates or unzips the two strands of DNA. This only occurs in the area of the single gene that's being transcribed. The rest of the chromosome stays connected in its double-stranded formation. So here we can see that happening. We might call this a transcription bubble. All right, so that's initiation. Now next we get elongation. So during elongation, only one strand of the DNA acts as a template for transcription. This strand is called the template strand, and it runs in the 3' to 5' direction. RNA polymerase reads that strand of the DNA, one nucleotide at a time, and builds an RNA molecule made out of complementary nucleotides. Like DNA polymerase, RNA polymerase can only build new strands in the 5' to 3' direction. That new RNA molecule that's built is going to be called the primary transcript. Think of it like a first draft. It carries the same sequence as the non-template or coding strand of the DNA, except that instead of having the nitrogenous base thymine, it has the nitrogenous base uracil, or U, instead. So wherever there would have been a T, there's a U instead in the RNA. Here we can see elongation happening. So the template strand of the DNA is being used to build the RNA. The other strand of DNA is sometimes called the coding strand. During transcription, it's just sort of hanging out over here, really not doing anything. Notice that we can see the RNA being made here. Eventually, we'll get to termination. RNA polymerase eventually gets to the end of the gene. At that point, a stop or terminator sequence in the DNA signals, hey, you're at the end, and RNA polymerase will stop transcription. The RNA molecule will get released, and the RNA polymerase will unattach from the DNA. The DNA strands that were unzipped will zip back together like nothing ever happened. Again, many times RNA polymerases are transcribing genes one at a time. And this allows the cell to produce many copies of the same mRNA in a very short amount of time. So here we can see the process being terminated. So next we get what's called RNA processing. So in prokaryotic cells, RNA processing doesn't happen. The RNA molecules that are made during transcription can go and be immediately translated and used to make proteins. But in eukaryotic cells, these newly formed RNA molecules have to go through this process of RNA processing. The RNA molecules, like we said before, that were originally made are called pre-mRNA molecules or primary transcripts. The process of RNA processing has 3 or 4 things that can happen. First of all, we get what's called front end or 5' modification. To the 5' end of the RNA, a protective cap is added. And this cap helps the RNA to later attach to the ribosomes. Then we get back end modification or 3' modification. Here, a long strand of adenine A nucleotides is added to the 3' end, and this helps to protect the RNA molecule from enzymatic breakdown, and it also helps it to be exported from the nucleus. But maybe the most important part of RNA processing is called splicing or middle modification. Eukaryotic genes contain both exons, which are the protein coding regions, and introns, which are non-coding regions. When transcription happens, both the exon sequences and the intron sequences are transcribed. During splicing or middle modification, those intron sequences are cut out and the exon sequences are spliced back together into a mature mRNA. And this is carried out by a set of enzymes called spliceosomes. Sometimes you might hear them called ribozymes, or related enzymes called ribozymes. Now, the important thing about this is that sometimes a DNA sequence that's treated as an exon in certain situations can be treated as an intron in another situation. So that allows one gene to be processed in different ways. That's why sometimes this process, you'll hear it called alternative processing. And what that ultimately ends up allowing to happen is it allows one gene to actually code for several different proteins. Here we can see some of that happening. So we can see the DNA has exons, the coding sequences, separated by introns. During transcription, all of that is transcribed. But during processing, these introns are removed and the exons are reconnected together. Like we said before, though, sometimes an exon can be treated as an intron, and that allows each gene to actually code for many, many different proteins. For example, we only have a handful of antibody protein coding genes, but because of this alternative processing, our body can produce, let's see, that would be how many? That would be about 1 trillion distinct different types of antibodies because of all the different alternative processing combinations that can happen. So this is a way to get extreme protein diversity. Here we can see just one gene. Here all the exons are used and we make this protein. In this situation, exon 3 is actually treated as an intron. So that leads to a different mature RNA, which ultimately ends up looking at a different protein. Here, exon number 4 is treated as an intron, and that gives us, again, a different mature mRNA and yet a different enzyme. And since structure determines function, all of these proteins have different structures and will ultimately have different functions. All right, so let's go ahead and talk just a minute about translation. So during translation, a cell essentially reads the information encoded in the mRNA molecule and uses it to build a polypeptide. A polypeptide is essentially a single chain of amino acids. Usually several of those polypeptides are wrapped together to make a large three-dimensional protein. Each set of 3 RNA nucleotides, called a codon, codes for one specific amino acid. There's 4 different types of nucleotides in mRNA, A, U, C, and G. Those can be combined into 64 different 3-base codons. 61 of these code for amino acids. One of these, the sequence AUG, codes for the amino acid methionine, but it also acts as like the start codon to signal the beginning of a protein, the beginning of protein synthesis. And the other 3 codons, those act as stop codons. They tell the cell that the polypeptide is complete and that it's time to stop translation. So scientists have decoded the genetic code. They know what it codes for, and they illustrate this code either in the table format or the wheel format. You need to be able to use either the table here to determine an amino acid sequence or the wheel located here. So in addition to the messenger RNA, another type of RNA called transfer RNA or tRNA play a role during the process of translation. tRNA molecules are shaped almost in a cross-like arrangement. Their job is to transport specific amino acids to the ribosomes and put them in essentially the right place in the newly built protein. On one side of the tRNA molecule is a set of 3 unpaired nucleotides called an anticodon. This essentially tells the tRNA where to go, where to park. And those tRNA anticodons have to pair up with a specific mRNA codon. Here we can see this happening. So here's this whole thing here is a tRNA molecule. It carries its amino acid up here, and this is its anticodon. So that anticodon has to be complementary to a codon in the mRNA or on the mRNA. So let's talk for a minute about ribosomes. We know ribosomes are the structures where translation takes place. They build the proteins. They're actually made of RNA and protein, a type of RNA mostly called rRNA, ribosomal RNA. They organize translation. They catalyze the reactions that actually join the amino acids together to make the polypeptides. As we've said before, many of them are embedded in the membrane of the roughy R. And these make proteins that are either exported out of the cell or that end up embedded in the cell membrane. There are also freely floating ribosomes. They freely float in the cytoplasm, and they make proteins that function within the cell that made them. Ribosomes are made out of two main parts, the large and small subunits. These two pieces are usually separate, but they come together around an mRNA molecule, and they form a fully functioning, complete ribosome during translation. Once translation is complete, the two subunits disassemble again. We'll see on the next diagram that each ribosome has 3 slots that we'll talk about for tRNA molecules. They're called the E, the P, and the A sites. So here we can see that, let's go to a different slide here first. So let's talk about the initiation of translation first. So like transcription, translation is said to occur in 3 stages: initiation, elongation, and termination. Initiation begins when the initiator tRNA, which carries the amino acid methionine, attaches to the small portion of the ribosome. Together, they bind to the 5' end of the mRNA by recognizing the 5' cap, the little protective cap on the end. They then walk along the mRNA in the 5 to 3 direction, stopping when they reach the start codon on the mRNA. That's the AUG. So here we can see that happening. So here's the initiator tRNA. Here's its anticodon. Here's the amino acid that it's carrying, methionine. It's attached to the small portion of the ribosome. Together they attach to the messenger RNA at the 5' cap, and they move their way down until they get to the start codon, the AUG codon on the mRNA. At that point, the large ribosomal subunit comes in and joins. We see that in green here. So that's initiation. Next, we get elongation. So during elongation, amino acids get added, and we start to build a polypeptide chain. The chain gets longer and longer. The original initiator tRNA begins the process of elongation in the P site of the ribosome. Another codon sits unpaired in the A site of the ribosome. Let's look at a picture. So here's the P site, and the A site would have been unoccupied to begin with. So what happens next is the next tRNA will come in. Its anticodon, in this case AAA, has to match the codon that was sitting in that A site on the ribosome, UUU. So it brings with it an amino acid. The large part of the ribosome catalyzes a reaction between these two amino acids. So they bond, they form a covalent peptide bond. And we see that happening here. The ribosome scoots over, and that initial tRNA now ends up in the E site. Think of that like the exit site. So that tRNA leaves. It leaves behind, though, the amino acid that was attached to it. It will eventually go and reload and be reused again and bring back another methionine. But essentially, now notice the A site is empty again here. So there's a codon GCU sitting in that A site. So the only tRNA that can park in this spot is one that has a complementary anticodon, in this case CGA. So it will come in, bind in that spot. This amino acid will get attached to the alanine, and then the whole complex will scoot down. the tRNAs that sit in the E site will exit, and they'll go and get another copy of that same amino acid they originally delivered to this one. So here we have a growing polypeptide chain. Eventually, we have termination. So this is when translation stopped, and it happens when the ribosome reaches a stop codon, one of these three. So it's recognized by some proteins called release factors. They fit into the P site of the ribosome. They cause the ribosome to add a water molecule to the last amino acid of the polypeptide chain, and this separates it from the tRNA, and that newly made polypeptide is released. The small and large subunits of the ribosome separate, and the tRNA molecules are freed. It's very likely that all the components will come back together again to make another copy of that same polypeptide. So here we can see termination happening. Now we will mention briefly the Wobble effect. Turns out that the third nucleotide of a codon doesn't require the same level of specificity as the first two in terms of binding. The third position can wobble between several nucleotide possibilities. Remember that there's 61 codons, but only 20 different actual amino acids. So some tRNA anticodons have this nitrogenous base called inosine, or inosine. I'm not sure how you pronounce it with an I, in the third position. This nucleotide can bind with both A, C, and U. Because it can do that, this provides some protection against mutations that happen in that third position. Because even if an RNA is supposed to have an A, but instead has a C, the same tRNA can still bind to it, which means the same amino acid gets put into place. So the protein doesn't change. So that's a good thing for us, this Wobble effect, because it helps to protect us from potentially disease-causing mutations. The mutation still happens, but it's a silent mutation because the amino acid sequence doesn't get changed, and that's a good thing. After translation, we get a process called post-translation modification that happens. Essentially, this is the process in which the proteins are folded. Remember that proteins have this three-dimensional shape that has to be in place for them to function properly. Most of the folding takes place in the ER, the endoplasmic reticulum. Some polypeptides, specifically those that are made by free-floating ribosomes, require the assistance of these proteins called chaperonins to help them to fold in a favorable environment, free of the influence of water. So also during post-translation modification, some other things can happen. Glycosylation, which is the adding of carbohydrates to the protein. This can happen either in the ER or in the Golgi. Lipidation, lipids can be added to the protein. Phosphorylation can happen, where phosphate groups are added. And also ubiquitination can happen, where this small protein called ubiquitin can be added to a larger protein, and it can mark it for degradation by the proteasomes in the cell. So let's stop there. So that we'll end our discussion of the central dogma there, and in our next screencast we'll talk a little bit about mutations.